What I'll do is be talking about Translarna and just, this is just forward-looking statements. We're a public company. This is just to remind you what our mission is to discover, develop, and commercialize new therapies for rare and neglected disorders. And this just shows you, in a sense, the timeline. I think I showed this earlier before, is we founded the company in 1998 and identified Atelorn or Translarna in 2003. And you can see from 2003 to where we are in 2017 of all the different trials and hurdles that were required. And this, we were early, as Pat had said, within this, where there really wasn't much natural history really known or any registration trial that came before us. So much of what we had to do really was, in a sense, what we always said is to build the plane and fly it at the same time. But we moved forward, and as you could see, we got in Europe the first approval for a Duchenne muscular dystrophy drug, conditional approval in Europe that was then redone just recently. So it's on the market, where we're now selling to companies around the world. And as you can see, we also have now an FDA PDUFA date of October 24th with a AdCom coming up in September 28th. So exciting times, not only just in Europe and other places in the world, but also now an opportunity to make our case in the US. This just shows you that with the nice thing with a European approval, it makes it available and accessible to many different countries. It's now commercially available in over 25 countries. You can see in the orange. It's not only in Europe, but also in Canada, South America, Europe, Middle East, and North Africa as well as other parts of Asia. So you see that we're now a company that really has been able to bring Translarna to patients around the globe. So what is Translarna? Let me just remind you, it's the goal is to treat those patients who have Duchenne, loss of production of dystrophin as a consequence of a mutation type called a nonsense mutation. And so what does that mean is, so if this is an example of an RNA, so it's DNA to RNA, the protein, the RNA is in a sense the message that has the information to make the dystrophin protein, and normally that message then goes on, makes the protein. In the case of a premature stop codon, what happens is you get a single base mutation that in a sense changes what should normally be an amino acid that's added on, and it makes it prematurely stop. It's very much analogous to being putting in a period in the middle of the sentence, so it's not completed, and therefore you lack the production of the protein. And that's why you lack dystrophin, and that's why you get Duchenne muscular dystrophy for this type of mutation. About 10% of the Atelorn works by binding to the ribosome, tricking the cellular machinery to continue on, honor the normal termination codon that's normally present in the cell, and therefore makes the full-length protein. And so we've done a lot of work and exploration over the last 2 decades, and we know that it produces dystrophin. It's highly specific for a nonsense mutation, but not normal termination codons, and it doesn't overlap, for instance, from the exon skipping or other things. So it's complementary to those patients who only have a nonsense mutation. This just shows you here is examples using either a Duchenne muscular dystrophy MDX mouse model or from a human human muscle cell that's grown, and you can see on the left is just a positive control, where we're looking for dystrophin. This is a myotube where we're looking for dystrophin protein. It's fluorescent green because you could see it's with fluorescein. You can see in the middle is what an untreated nonsense mutation muscle looks like. It's virtually devoid of dystrophin. And then treatment with adalurin allows dystrophin to be made, and you can see it quite nicely. Also, as you have seen in the MDX mouse, it's again, adalurin is an oral drug, and you could see a normal control in the middle. You could see the mouse MDX nonsense mutation, and it's the TA diaphragm and heart, so it's fast muscle, slow muscle, and cardiac muscle. And then you treat with adalurin. You could see, again, at the production and localization of the molecule of dystrophin to the right place in the membrane. Importantly, also, you can look like almost going down steps. So you could do, in a sense, eccentric contraction injury that lets you look and see if dystrophin is doing the job it's supposed to be doing. And you could see that in a normal mouse, very little injury occurs. In a MDX mouse, there's far more injury, on the order of 30%. And after treatment with adalurin or Translarna, it's able to prevent that injury. So we knew that it's being made, that it's localized to the correct place, and is functional. We've also, then from there, from this work, we went to the phase 2 study, and this was a short-term study where we treated Duchenne patients for 28 days with Translarna, and then took the extensor digitorum brevis muscle from them either pre or post, and then monitored dystrophin production in those patients afterwards. And the nice thing about the EDB is that it's a small little muscle on the toe, so you can take actually the whole muscle and therefore really be able to look at it quite well. So you can see here, we're able to see increases of dystrophin. Now, really, when you do it in patients, I think what the community has learned is that there are significant challenges in doing biopsies, not only because dystrophin's just a damn hard molecule to see, it's one of the largest molecules and hard to actually quantitate, but also there's sampling challenges, and that within a muscle, there could be more fat or less fat within a piece that you take, and therefore you can get sampling issues that you may have taken a piece in one part of the muscle versus the other that can actually give you different results. So you have the complications of sampling and also technical difficulties of actually doing the assays in a real world environment. And so you really can only do this in really small sites where they're experts in it. And we found to try and do it in larger sites like what we did in Study 007, when you have 37 sites and you have to ship to multiple places, it's far more challenging to do. But the important point here is that we're capable of showing that we had a proof of concept and capable of seeing dystrophin being produced. And from there, what we wanted then to do is then, I should also say, so what work we've done, we've shown that it's able to promote read-through of a nonsense mutation in dystrophin. It could be a dose response. It doesn't read through normal termination codons. There's now over 40 publications in different nonsense mutation disease models demonstrating adalurin's activity. And then so we moved it into patients from there based on these proof of concept studies. And there is where we had to begin to think about what sort of tests that we could use to demonstrate the differences. And while, and you got to think about this now in the 2003, 2004, 2005 time frame, where there wasn't very much natural history done. And so we knew, and this just shows you many of the functional milestones that you monitor and can see where certainly Duchenne boys early on could lose the ability to get off the ground, get up steps. We know loss of ambulation. So we know all of these, but how do you measure it in clinical trials? And over the years, it became clear that you want to look at a number of things. And so we initially set up the 6-minute walk test, got the regulators to understand that and be able to use that as an asset that they can approve drugs on. And also now the time function tests are important. The North Star Ambulatory Assessments turned out to be important. That measures 17 different functions. And these are ambulatory measures that you can look. Then you could also monitor the loss of ambulation. You can also monitor pulmonary function in those boys that are non-ambulatory. So this is the results I'm showing you here from 2 placebo-controlled trials. So they're randomized controlled trials here, well-controlled. And so from 2 different trials, on the left, the blue is study 007, the orange is study 20. And in both cases, we saw improvements in the 6-minute walk test as seen on the left. And then in the time function tests are the 10-meter run walk, the 4-stair climb, 4-stair descent, and you're comparing it to placebo. So you could see anywhere we saw from 15 to 32 meters in the 6-minute walk test. So we saw that the DMD boys walked better. And then you can see also in the time function tests, in all of those, you can see improvements in all of the time function tests. And so and these are, we think, are clinically relevant because they're in the where others have now shown that these are clinically meaningful differences. These tests are done relatively quickly in 6 to 10 seconds, and so 1 to 1.5 seconds, and we even saw yesterday that even less may be more clinically meaningful. And so we're seeing substantial improvements in that. The other interesting that we monitored was in the North Star Ambulatory Assessment. And here there's 17 different tests that are monitored. And you get a score of 2, 1, or 0. And so what you can see is 2 is you're easily able to perform the test. 1, you can perform it with difficulty, and 0 is unable to perform the test. And so, we looked at this data and we said, the difference between 2 and 1, it can be qualitative, whether depending on what person looks at it. But the loss of the ability to do it, the unable to go from being able to do it to unable to do it, measures a functional milestone and a loss of a functional milestone. And so can we see a difference in the loss of functional milestones when patients are treated with Adalurin? And so I think what you could see here is, again, the placebo is in gray and Adalurin is in orange, and I'll walk you through all the different ones. And you can see on the whole, there's less patients that lost these functional milestones, starting with some of the easier ones, where more patients on placebo lost them. And that's true as you go on to even more difficult, and then even more difficult. No matter where you looked and whichever functional milestones that were observed, Adalurin reduced the risk of losing that. And there was a 31% reduction in the risk of that we saw. So what that really says is that you're preserving function. And ultimately, that's what I think what we all care about, is to preserve function in patients. The other interesting, we've had patients on, and the number of have come up on Adalurin over 10 years, and we have a number of patients who were non-ambulatory, who we monitored pulmonary function. And when you look at pulmonary function, like whether you have Duchenne muscular dystrophy or us, you improve in lung function over time, and then you start declining over time. And you can see we used the Synergy database, a natural history database, matched patients from that database to patients that were in the non-ambulatory portion of study 19, and compared pulmonary function in those patients. And what you could see is that, and it's really quite striking, that there's, in the natural history data, you could see that somewhere 12 and a half years of age, that there's a change in the slope where they're beginning the decline in pulmonary function. If you look at the Adalurin treated patients, you're seeing the slope change occur at 16.5 years. And we think this is really quite promising data because it's saying not only are you monitoring in patients that are where we could see the effects in ambulatory patients, but we could also now see it in non-ambulatory patients as well. So, so this is some of the data that we have that we've built over the years and demonstrating Adalurin's efficacy over the years. And we have a number of other trials that are occurring. As I said, it's approved in Europe. One of the specific obligations is to do a multi-center long-term study of Translarna. It's going to be a long-term study looking at long-term outcomes. We also have a pediatric study for 2 to 5-year-olds that we've completed, PK studies that have been done. So those have been completed. We've also had extension trials, 19 and 20E, that have been going on in the US. 19 is probably close to 10 years, where we're going to be transitioning those patients to a new protocol, to 016, so they'll all be getting the drug, but it won't be in the same amount of measurements in terms of the clinical trial, so we think it'll be easier for patients to be on. We have already long-term data on that. We also, as part of the commitment in getting approval in Europe, and this is just very common for most orphan drugs where you continue to study it, even in a commercial setting. So we have a registry that captures both, importantly, safety, but also efficacy measurements. And if we get approval in the US, we'll continue to move that registry into the US as well. So that's where we are in terms of Adalurin, in terms of the trials. We continue to focus in multiple areas in dystrophin restoration. We're obviously working on an alternate formulation on this, perhaps a chewable capsule. We have next generation compounds that can enhance Adalurin, things that are different chemically, to continue to build on that. And we have programs in inflammation and muscle regeneration that also. So there's a real, obviously we've had a nearly 20-year commitment in this field and will continue to do so. Then the important point here is that we have our day in court. The PDUFA date for Translarna is October 24th of this year, with the advisory committee meeting set tentatively, which I think will be less tentatively as days go on, for September 28th. So I think, I hope this has given you a taste of the process of getting a drug to be able to get it commercially approved. And we often draw, you start and it's a straight line and you end and finish. But that's sort of the gloss. The reality is it's a winding road, and that and there's fits and starts and stops, and that's because at the end, and this is very important for the community to understand, because in a way you're still doing science, especially in areas where it's less well understood, and so it really takes from an idea to a drug somewhere between 15 and 20 years, and I think the community, it's important for the community to realize this. You have 2 drugs that have been approved, really, and that actually shows you that we've beaten the odds. I mean, if you look, it is on the average a 10 to 20 year period if it's from an idea to a drug. And this just shows you the success rates of that. And so, and I think it's good to talk about this. It's hard. From idea to a drug is about a 95% chance it won't make it. So the fact that some drugs in this community have already got there is exciting. And the fact that you saw what Pat put up, all those trials, says that something could win. We don't know what, but something will win. And this just, I think, points out nicely that, because there's always a lot of hype in biotech, but from 1950, this is from 2009, from 1950 till now, there's been about 4,300 biotechs. The number of biotechs that then brought drug to product is 261. So it shows you that this group is in rarefied air, where drugs have gone through the process, ideas have been tested, and drugs have been brought to the marketplace. And that's in a part of the effort, I think, of the whole community, because without people like Pat and everyone here, and just pushing and getting it, it's in good stead. Think about a patient population, 9,000, has all those trials that are occurring. It's really quite exciting. So we're proud to have brought the first drug through approval, and hopefully we'll have the opportunity to bring other ones as well. And as I say, it's very important to do this, so even though the odds are against us, I think all of us here plan to do our best and be optimistic to make it happen. So with that, let me thank you.